Gene therapy is when DNA is introduced into a patient to treat a genetic disease. The new DNA usually contains a functioning gene to correct the effects of a disease-causing mutation. Gene therapy uses sections of DNA, usually genes, to treat or prevent disease. The DNA is carefully selected to correct the effect of mutated gene that is causing disease. The technique was first developed in 1972 but has so far had limited success in treating human diseases. Gene therapy may be a promising treatment option for some genetic diseases, including muscular dystrophy and cystic fibrosis. Genetic diseases, a disease that is caused by a change in an individual's DNA, also known as a genetic disorder. Changes or mutations that occur in the DNA sequence of a single gene cause this type of inheritance. There are thousands of known single gene disorders.
Cystic fibrosis is a genetic disease. People with CF have inherited two copies of the defective CF gene, one copy from each parent. Both parents must have at least one copy of the defective gene. People with only one copy of the defective gene are called carriers, but they do not have the disease. Alpha and beta thalassemias. The thalassemias are a group of inherited hematologic disorders caused by defects in the synthesis of one or more of the hemoglobin chains. Alpha thalassemia is caused by reduced or absent synthesis of alpha globin chains, and beta thalassemia is caused by reduced or absent synthesis of beta globin chains. Sickle cell anemia. Sickle cell anemia is an inherited form of anemia, a condition in which there aren't enough healthy red blood cells to carry adequate oxygen throughout your body. Normally, your red blood cells are flexible and round, moving easily through your body vessels. In sickle cell anemia, the red blood cells became rigid and sticky and are shaped that like sickles on a crescent moon. These irregularly shaped cells can get stuck in a small blood vessels, which can slow or block blood flow and oxygen to parts of the body.
Marfan syndrome is a genetic disorder of connective tissue. The degree to which people are affected varies. People with Marfan tend to be tall and thin, with long arms, legs, fingers, and toes. They also typically have flexible joints and scoliosis.
Scoliosis. Fragile X syndrome is a genetic condition that causes a range of development programs, including learning disabilities and cognitive impairment. Usually, males are more severely affected by this disorder than females. Affected individuals usually have delayed development of speech and language by age of 2. Huntington's disease is a fatal genetic disorder that causes the progressive breakdown of nerve cells in the brain. It deteriorates a person's physical and mental abilities, usually during their prime working years, and has no cure. Hemochromatosis is a disorder where too much iron builds up in your body. Sometimes it's called iron overload. Normally, your intestines absorb just the right amount of iron from the foods you eat. But in hemochromatosis, your body absorbs too much, and it has no way to get rid of it. killing of specific cells, suitable for diseases such as cancer that can be treated by destroying certain groups of cells. The aim is to insert DNA into the cells that causes the cell to die. This can be achieved in one of two ways. The inserted DNA contains a suicide gene that produces a highly toxic product which kills the diseased cell. The inserted DNA causes expression of a protein that marks the cells so that the diseased cells are attacked by the body's natural immune system. It is essential with this method that the inserted DNA is targeted appropriately to avoid the death of cells that are functionally normally. How is DNA transfer done? A section of DNA gene containing instructions for making a useful protein is packaged within a vector, usually a virus, bacterium, or plasmid. The vector acts as a vehicle to carry the new DNA into the cell of a patient with a genetic disease. Once inside the cells of the patient, the DNA gene is expressed by the cell's normal machinery, leading to production of the therapeutic protein and treatment of the patient's disease. Challenges of gene therapy: delivering the gene to the right place and switching it on. It is crucial that the new gene reaches the right cell. Delivering a gene into the wrong cell would be inefficient and could also cause health problems for the patient. Even once the right cell has been targeted, the gene has to be turned on. Cells sometimes obstruct this process by shutting down genes that are showing unusual activity. Avoiding the immune response. The role of the immune system is to fight off intruders. Sometimes, new genes introduced by gene therapy are considered potentially harmful intruders. This can spark an immune response in the patient that could be harmful to them. Scientists, therefore, have the challenge of finding a way to deliver genes without the immune system noticing. This is usually by using vectors that are less likely to trigger an immune response. Making sure the new gene doesn't disrupt the function of other genes. Ideally, a new gene introduced by gene therapy will integrate itself into the genome of the patient and continue working for the rest of their lives. There is a risk that the new gene will insert itself into the path of another gene, disrupting its activity. This could have damaging effects. For example, if its interface in an important gene involved in the regulating cell division, it could result in cancer. The cause of gene therapy. Many genetic disorders that can be targeted with gene therapy are extremely rare. Gene therapy, therefore, often requires an individual, case-by-case approach. This may be effective but may also be very expensive.
Hi, ano nga ba ang genes therapy? Ang genes therapy ay isang uri ng treatment na kung saan hindi tayo gumagamit ng medicine drugs. Kumbaga, ginagamit natin ang sariling cells upang malunasan or ma-prevent natin ng iba't ibang sakit. Ito ay gumagamit lamang ng cells or ng genes ng ibang tao o sarili natin genes. Yun po ang genes therapy para sa akin. Salamat.
Ang pagkakaalam ko sa gene therapy stem cell ay tungkol sa DNA. Tinutukoy ng iba't ibang uri ng stem cell diseases at paano binibigyan ng solusyon gamit ang gene therapy.
Gene therapy is an experimental technique used by the doctors to cure, to treat, or to prevent diseases. So there are two types of gene therapy, according to which type of cells are going to be treated. First one is the somatic gene therapy, and the last one is the germline gene therapy. So by means of this technique, doctors be able to treat disorders by inserting a gene into a patient's cells. So instead of using drugs or performing a surgery.